Next one, histones. Okay, here start. Histones are rich in, okay, what? Histones are rich in amino acids, that is lysines and arginines. Histones organize unit of 8 molecules, okay, that are arranges in the 8 molecules. 1 2 3 4 5 5 6 7 8, okay, backside 1 8, that 8 is called as octamer, okay. DNA negatively charged wrapped around positively charged histone octamer to form a structure called nucleosome, okay. The typical nucleosome having a 200 base pairs of a DNA helix, okay. Here when we call this, okay, this it contains 200 base pairs of DNA. Nucleosome is the repeating unit of structure in a nucleus that we call as chromatin, okay. Chromatin thread like strained body in nucleus. Nucleosome in chromatin sees as beds, okay, beds on string in the electron microscope. In my electron microscope it looks like a beds on a string, okay. Here beds on a string. Beds on a string structure, chromatin packaged chromatin fibers further coiled and condensed during metaphase chromosome. Packaging of chromatin requires additional set of protein that we know called as non-histone chromosome, NHC protein. Why? Because of package of chromatin requires additional set of protein. Typical nucleus region of chromatin loosely packed euchromatin, transcriptionally active. Chromatin densely packed heterochromatin inactive. Okay, heterochromatin is nothing but inactive, and that of chromatin loosely packed, densely packed heterochromatin, loosely packed euchromatin. And what is the transcriptionally active chromatin means euchromatin. Okay, inactive means heterochromatin. And difference between in the board, the very important question of difference between euchromatin and heterochromatin. Search for a genetic material, okay, is the history how the first calculate that DNA is the genetic material and not RNA, okay. And there are some method, okay, there are some experiment of which is the genetic material, whether it is a DNA or RNA. It is a contradictory in the that year, 19th century. Therefore there are some scientists, they come up with an experiment and they gave their idea, okay. One is very important, that is transforming principle, that was given by Frederick Griffith, okay. Principle or factor responsible for heredity in organism. In 1928, FG means Frederick Griffith series of experiment, okay. He did a series of experiments on the Streptococcus pneumonia. It is very important. Griffith means Streptococcus pneumonia, which causes pneumonia, okay. Observed changes in the physical form of bacteria. That is very important. He observes change in physical form of bacteria transformation, and he called, because of the change in physical form, he called as transforming principle, okay. And first, Streptococcus pneumonia grown in culture plate, okay. In culture plate, 2 different colonies observed, okay. He observed 2 different colonies, one smooth colony and another one is rough colony. What do you mean by smooth colony? Means nothing but S strain, okay. Mucus polysaccharide coat, okay. Mucus polysaccharide coat, which is virulent, okay. The virulent which causes pneumonia, okay. Then rough colony means R strain, okay. Does not have mucus coat, non-virulent, which does not causes pneumonia, okay. This was the main difference, okay. What he do, he cultured both, okay, 2 colony, that is S strain and R strain. In S strain he injected, okay, infected or injected to mice S strain, okay, and the mice died due to pneumonia, okay, and he also injected R strain to the mice and that will survive, okay, and he conclude that when S strain, which is virulent, that is the coat, okay, mucus coat, injected into mice, the mice died due to the pneumonia, and that of R strain does not have a mucus, okay, that is a polysaccharide coat. When injected into mice is alive, and he concluded that protein, okay, in next